This is Trivalika and today I am here to discuss about the isolation of plant genomic DNA by CTAB method. The aim is to isolate the genomic DNA from plant cells by CTAB method. The materials required are green leaves, vials, tissue grinder, ammonium acetate, incubator, centrifuge, TE buffer, isoamyl alcohol and isopropanol. First, we need to prepare the CTAB buffer. In order to prepare 100 ml of CTAB buffer, we need to add 1 gram of CTAB powder, 10 ml of 1M Tris pH 8, 4 ml of 0.5 m EDTA, 28 ml of 5 m NaCl and 40 ml of distilled water. Then we need to adjust to pH 5 with HCl and make up to 100 ml with distilled water. The principle of this experiment is the cell is disrupted physically or chemically to get a fluid containing all the cell components including DNA. During cell lysis, different chemicals and reagents are used to break down different cell components. Example, lipids are broken down by detergents and surfactants. Proteins are broken down by protease and RNA is broken down by using RNase. Then lysate is treated with concentrated salt solution to make the broken component clump together and leave the DNA freely floating in the solution. This solution is centrifuged to separate the clump debris from DNA. At last, DNA precipitation is done by adding ice-cold alcohol plus a salt to increase the ionic strength, which increases the precipitation process. A pellet of DNA is obtained about centrifugation of this solution. Supernatant is discarded except for the DNA pellet, which remains stuck to the walls of the append-off. Pellet is then suspended either in slightly alkaline solution, mostly TE buffer or ultra-pure water for subsequent DNA experimentation. The procedure is, first we need to collect a fresh young green leaves and clean with sterile distilled water. Then we need to weigh an amount of 25 mg and collect into vial. We need to grind the material with the help of tissue grinder in 400 ml CTAB buffer under cool conditions. We need to incubate the vial at 60 degrees Celsius for 30 minutes, occasionally mixing with gentle swirling. Then centrifuge at 15,000 rpm for 5 minutes and collect the supernatant into a clean tube. Then we need to add equal volume of chloroform, isoamyl alcohol and mix in 24:1 ratio and 50 ml of ammonium acetate to it. Then invert the tube for 5 times and centrifuge at 15,000 rpm for 5 minutes. After that, collect the upper aqueous layer into a clean tube without disturbing the interface layer. Here you can see the upper aqueous layer and the organic layer. Now, reprecipitate the DNA by using double the quantity of chilled ethanol or isopropanol. Then centrifuge the content to precipitate the DNA. Discard the supernatant and air dry the pellet. Add 50 ml of TE buffer and vortex to mix the content in the buffer. After that, dilute the DNA 1000 times in TE buffer and quantify by taking the OD at 260 nanometer with a spectrophotometer. Take readings at 260 nanometer by 280 nanometer ratio as an indicator of DNA purity. Then observe the purified DNA on 1% agarose gel after staining with ethidium bromide. Now, as a result, we can observe this isolated DNA on 1% agarose gel. Thank you guys, and if you have any doubts, please let me know in the comment section.